This is Dr. Vishnu Shankar Sinha, PG Department of Botany, Tata College, Chaivasa. Today I am going to discuss the Krebs cycle or tricarboxylic acid cycle. Krebs cycle is the aerobic respiration of eukaryotic cells. The citric acid cycle or tricarboxylic acid cycle is a part of cellular respiration. It was first elucidated by a German biochemist, Sir Hans Adolf Krebs, and the Krebs cycle named after Hans Krebs. The Krebs cycle playing a starring role in both the process of energy production and biosynthesis. Krebs shared the Nobel Prize for Physiology and Medicine in the year 1953 with the Fritz Albert Lippmann, the father of ATP cycle. Krebs cycle is the central metabolic hub of the cell. The site of Krebs cycle in eukaryotic cell is inside mitochondria, that is mitochondrial matrix, and glycolysis takes place in the cytosol. This picture clearly showed the Krebs cycle is the common pathway for the oxidation of carbohydrates, proteins, and fat. The main function of the Krebs cycle is to produce electron carriers that can be used in the last step of cellular respiration. Now, the two-carbon compound acetyl coenzyme A is the starting point for the Krebs cycle, and the acetyl coenzyme A is formed from the three-carbon compound pyruvic acid by citrate pyruvate dehydrogenase and release one molecule of carbon dioxide and one molecule of NADH2. This is the reaction where the two-carbon compound acetyl coenzyme A is synthesized from the three-carbon compound pyruvic acid in the presence of pyruvate dehydrogenase and release one molecule of carbon dioxide and one molecule of NADH2. Now this is the overall reaction of the Krebs cycle where the acetyl coenzyme A is oxidized into carbon dioxide and water. Yes, this is the entire reaction of the Krebs cycle, and it can be easily understood in 8 reactions, and the intermediate compounds of the Krebs cycles may be 6 carbon compound, that is citric acid, isocitric acid, and 5 carbon compound, that is alpha ketoglutarate acid, 4 carbon compound, that is succinyl coenzyme A, succinic acid, fumaric acid, malic acid, and oxaloacetic acid. In the Krebs cycle, there are so many intermediate compounds are biosynthesized from the intermediates of Krebs cycle. That is, the amino acid purine and pyrimidine are synthesized from the aspartate. Porphyrines, heme, and chlorophyll molecules are synthesized from the succinyl coenzyme A. Similarly, purine is synthesized from the glutamate. The fatty acid esterol is also synthesized from citric acid, and one molecule of NADH2 oxidation produces 3 molecule of ATP, and one molecule of FADH2 produces 2 molecule of ATP. And in one Krebs cycle produces 3 molecule of FADH2, 1 molecule of FADH2, and 1 molecule of ATP, and liberate 2 molecule of carbon dioxide. This is the reactions of Krebs cycle. Reaction number 1, formation of citrate. The 4 carbon compound oxaloacetic acid combined with the 2 carbon compound acetyl coenzyme A synthesizes 6 carbon compound, that is citric acid, in the presence of citrate synthetase and liberate coenzyme A. In reaction number 2, formation of isocitrate. The 6 carbon compound citric acid is converted into isocitric acid in the presence of aconitase enzyme and liberate 1 molecule of water. Isocitric acid is converted into 5 carbon compound alpha ketoglutarate acid and liberated 1 molecule of carbon dioxide and 1 molecule of NADH2 in the presence of isocitrate dehydrogenase. In reaction number 4, oxidation of the 5 carbon compound alpha ketoglutarate acid to 4 carbon compound succinyl coenzyme A in the presence of alpha ketoglutarate dehydrogenase complexes and also liberate one molecule of NADH2. In reaction number 5, conversion of succinyl coenzyme A to succinate in the presence of succinyl coenzyme A and liberate coenzyme A. Reaction number 6, oxidation of succinic acid to fumaric acid. The 4 carbon compound succinic acid is converted into 4 carbon compound fumarate in the presence of succinate dehydrogenases and liberate one molecule of FADH2. In reaction number 7, the fumaric acid is converted into malic acid in the presence of fumarase and involve of one molecule of H2O. In reaction number 8, oxidation of malate to oxaloacetic acid or generation of oxaloacetic acid. The 4 carbon compound malic acid is converted into oxaloacetic acid, that is also a 4 carbon compound in the presence of malate dehydrogenase and liberate one molecule of NADH2. Regulation of citric acid or TCA cycle. The concentration of ATP and NADH citrate acetyl coenzyme A primarily regulate the TCA cycle or citric acid cycle. The low energy state indicator molecules are ADP, AMP, NAD, stimulate tricarboxylic acid cycles. Enzymes, that is isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Isocitrate dehydrogenase is allostatically and stimulated by ADP, which enhances the enzyme's affinity for substrates, thus they are the key control point. Amphibolic. TCA or tricarboxylic cycle plays both catabolic and anabolic role. The catabolic role is oxidation of acetyl coenzyme A to carbon dioxide and water, that is H2O, giving out energy. The anabolic role of TCA cycle is synthesis of heme formation, FS synthesis, cholesterol, steroid, etc. from the intermediates of tricarboxylic acid cycle, as I have already discussed. Significance of the Krebs cycle: complete oxidation of acetyl coenzyme A. Number 2, amino acid, nucleotides, chlorophyll, cytochromes, fat, etc. biomolecules are synthesized from the compounds of Krebs cycles. Succinyl coenzyme A take part in the formation of chlorophyll molecule. Alpha ketoglutarate acid, pyruvic acid and oxaloacetic acid synthesizes amino acids. As provider of the energy, that is adenosine triphosphate, required for the various metabolic activities of the cell. By this cycle, carbon skeletal are got which are used in process for growth and for maintaining the cell. Thank you very much for watching. For detail, you may consult the biochemistry of Harper and etc. Thank you.